The cerebellar cortex is gray matter surrounding central white matter and is organized into three distinct layers, uniform throughout the cerebellum. The molecular layer is the most superficial, lying just beneath the pia mater. It contains dendritic arbors of Purkinje cells, parallel fibers from granule cells, and inhibitory stellate and basket cells. This layer is primarily a site of synaptic integration. Beneath it lies the Purkinje cell layer, a single row of large Purkinje neurons. These cells form the only efferent output of the cerebellar cortex and project inhibitory signals to the deep cerebellar nuclei and vestibular nuclei. The deepest layer is the granular layer, densely packed with granule cells and Golgi cells. Granule cells receive excitatory input from mossy fibers originating from spinocerebellar and pontocerebellar tracts. One of the most important cerebellar tumors is medulloblastoma, a common infratentorial pediatric neoplasm arising from the granular layer of the cerebellar cortex. Their axons ascend to the molecular layer and bifurcate into parallel fibers. Deep to the cortex lie the deep cerebellar nuclei, embedded within white matter. From medial to lateral, these are the fastigial, globose, emboliform, and dentate nuclei, remembered by the mnemonic "Don't eat greasy food." The fastigial nucleus, associated with the vermis, projects to vestibular nuclei and ventral lateral thalamus to influence axial and proximal muscles. The interposed nuclei project via the superior cerebellar peduncle, decussate, and influence the red nucleus and thalamus with final effects on ipsilateral limb muscles due to double decussation. The dentate nucleus projects via dentatothalamic and dentatorubral tracts, influencing premotor and motor cortices and forming feedback loops with the inferior olivary nucleus. All cerebellar output exits primarily through the superior cerebellar peduncle, while afferent input enters through the inferior and middle cerebellar peduncles.